Winter is coming in the northern hemisphere, and public health officials are asking how the seasonal shift will impact the spread of SARS-CoV-2, the virus that causes COVID-19. A new study tested how temperatures and humidity affect the structure of individual SARS-CoV-2 virus-like particles on surfaces. They found that just moderate temperature increases broke down the virus structure, while humidity had very little impact. In order to remain infectious, the SARS-CoV-2 membrane needs a specific web of proteins arranged in a particular order. When that structure falls apart, it becomes less infectious. The findings suggest that as temperatures begin to drop, particles on surfaces will remain infectious longer. This is the first study to analyze the mechanics of the virus on an individual particle level, but the findings agree with large-scale observations of other coronaviruses that appear to infect more people during the winter months. You would expect that temperature makes a huge difference, and that's what we saw. To the point where the packaging of the virus was completely destroyed by even moderate temperature increases, said Michael Verschinen, assistant professor at the University of Utah and co-senior author of the paper. What's surprising is how little heat was needed to break them down, surfaces that are warm to the touch, but not hot. The packaging of this virus is very sensitive to temperature. The paper published online on November 28, 2020, in the journal Biochemical Biophysical Research Communications. The team also published a separate paper December 14, 2020 in Scientific Reports describing their method for making the individual particle packaging. The virus-like particles are empty shells made from the same lipids and three types of proteins as are on an active SARS-CoV-2 viruses, but without the RNA that causes infections. This new method allows scientists to experiment with the virus without risking an outbreak. The SARS-CoV-2 is commonly spread by exhaling sharply, e.g. sneezing or coughing, which ejects droplets of tiny aerosols from the lungs. These mucus-Y droplets have a high surface-to-volume ratio and dry out quickly, so both wet and dry virus particles come into contact with a surface or travel directly into a new host. The researchers mimicked these conditions in their experiments. They tested the virus-like particles on glass surfaces under both dry and humid conditions. Using atomic force microscopy, they observed how, if at all, the structures changed. The scientists exposed samples to various temperatures under two conditions, with the particles inside a liquid buffer solution, and with the particles dried out in the open. In both liquid and bare conditions, elevating the temperature to about 93 degrees Fahrenheit for 30 minutes degraded the outer structure. The effect was stronger on the dry particles than on the liquid-protected ones. In contrast, surfaces at about 71 degrees Fahrenheit caused little to no damage, suggesting that particles in room temperature conditions or outside in cooler weather will remain infectious longer. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.